Hello students, this is Mr. Russell. I hope you enjoyed your Thanksgiving. I'm here today to talk to you about mitosis. And on the mitosis notes, which look like this, you will be filling out this side. And if you pay attention, most of the things that go in these blanks are going to be underlined in the PowerPoint that's in this video. So it should be real easy to just pause it whenever you get to something underlined and add it here. Okay, so you should have this side filled out already. That was just basic cell reproduction stuff, and this is the mitosis stuff that we're doing now. Okay, so last time Mr. Messina finished with the difference between somatic and sex cells. Okay, sex cells are your sperm and your egg. They're cells that are used for sexual reproduction. That's why we call them sex cells, also called gametes. Okay, not pronounced gametes or gametes, gametes. All right. So any cell in your body that's not used for sexual reproduction, so those could be the ones in your muscle, skin, neurons, which is in your nervous system, or anywhere else, those are all somatic cells. And we're talking about somatic cells today because they use the process of mitosis to reproduce. Okay, so mitosis, as I just said, is a process where somatic cells divide, and this is for growth and repair. So you are a multicellular being with, I don't know, somewhere between 50 and 100 trillion cells probably. Okay, and you make more of those cells by growing. Okay, so the reason you are larger than you were the day you were born is because you have made many, many more cells. It's not the same cells that just keep getting bigger and bigger. Cells can only get so big before they have to divide into two. Once cells get over a certain size, they don't function properly and they have to split or else they will die. Okay. The other purpose we use mitosis for is to repair. Sometimes cells die, sometimes they become injured, and they have to be repaired. If you ever get a cut or a scrape on your skin and over time you see that start to heal, what you're seeing there is mitosis actually happening. The cells that are still there and alive, they will reproduce themselves to replace those ones that were damaged or removed during that injury. Okay, so very important. Growth and repair are the two main purposes, of mitosis. Okay, so very first thing that has to happen before anything can divide is you have to replicate your DNA. We talked about that last objective. All right, and that's where we take one double helix of DNA, the twisted ladder, we split it in half, and then we copy each side so that we end up with two identical strands of DNA. Okay, so we have to have two identical copies of the DNA that can go into each new cell before we can divide the cells. So that's the first step. And that's actually not really part of mitosis. That happens before mitosis starts during interphase. Okay? But what happens is those replicated chromosomes where you get the X shape where they're attached together kind of like this, they are actually going to get pulled apart so that one copy goes to each side and then they end up in each new cell. Okay, what you end up with at the end of mitosis are two daughter cells. Why they're daughter cells and not son cells, I don't know. That's just the way the world works, guys. Sorry. But we end up with two daughter cells that are produced from that one original parent cell. Also very important is the daughter cells are identical to the parent cell. So this is basically cloning. We're not making new interesting organisms here. We're just making more cells that were exactly like the original, or at least very close to it. Here's a little diagram that shows you how the process works. We start out with one parent cell, and then over time, there's some steps that come in the middle that we're going to discuss on the next slide or two. But what happens is you divide that one cell into two new cells, and they are identical. You'll notice you can see we just have 4 little chromosomes floating around in this one. Down here you can see we still have 4, but they're that familiar X shape because they have replicated. After they replicate, they will split, and then you'll see down here each new cell ends up with 4. So they're both now identical to one another and identical to the original cell. Okay, so there are 4 parts of mitosis. They do not include interphase, which is where the chromosomes are replicated, or also called duplication. You'll see that word used sometimes. So the DNA gets copied then, and then we go through these 4 phases, which I like to call PMAT: prophase, metaphase, anaphase, telophase. Okay, prophase, metaphase, anaphase, and telophase. And real briefly, basically what happens is Prophase, the chromosomes condense into that familiar X shape. Normally they're just like a big bowl of spaghetti floating around in the nucleus. Well, they have to condense into the familiar X shapes in order for us to separate them. And also your nuclear membrane is going to disappear here. During metaphase, the chromosomes, those X-shaped chromosome pairs, are going to line up across the middle of the cell. Looks kind of like the line of scrimmage in football. They're going to line up there. Anaphase, we actually separate them, so we're going to take that X and we're going to rip it apart. And then during telophase, that's really just the opposite of prophase, and we basically get the cell back to normal. And then we also will go through cytokinesis, which is where the cell actually rips in half. Okay. At the end, you end up with, it's still one cell, but it's got two new nuclei in it, each, and that's the plural of nucleus. So each of these two new nuclei are going to have the exact same DNA in them. Here's kind of a little slide that shows you how it works. Prophase, as I said, the chromosomes condense, nuclear membrane disappears. Sometimes people describe it and they'll add different phases, like they put pro-metaphase, which just means before metaphase. But you can see in metaphase here, they've lined up across the middle. Anaphase, they get ripped in half. Telophase, your new nuclear membranes form, and you can see the cell is starting to pinch off here in the middle. Okay, and that's part of cytokinesis, where we actually divide the cell in half. Okay. Let me talk about that a little more. It's where the cytoplasm and the cell membranes of that cell divide to make two new cells. And this is where you go from one cell with two identical nuclei to two totally separate cells. Looks kind of like this. You'll see you start to get these dents in the cell when it's starting to pinch. This is actually called cell cleavage. No, that's not a joke. Cleavage is actually a technical term for when two things split. Okay, so that's called cell cleavage, and here cytokinesis is complete, and you'll see we have two cells that are identical to one another. This is an actual electron micrograph, meaning a picture taken with an electron microscope. They've added color to it so you can see things going on, but you can see this is right before the very end of cytokinesis because it's almost totally pinched off there in the middle. Okay, so that's mitosis. It's pretty simple. Important thing to remember is that you have to replicate your DNA first, so you have two identical copies, and then during mitosis, each replicated chromosome gets ripped in half so that each new cell ends up with one exact copy. So at the end, we end up with two identical cells, and they are identical to each other and to the parent cell. And we use it for two things, mainly it's growth and repair. This is not how you make new organisms, at least not how we humans do it. It's how we make more cells for ourselves as an organism. All right, thanks. If you have any questions, write them down so you can ask us in class. And I hope you guys enjoy the rest of your break.